Good morning, it's 9 AM here in San Diego, so we will start today's webinar. Today's webinar is on how to embed fully interactive 3D molecules into a web page or into PowerPoint. My name is Andrew Ori. You can email me or call after the webinar, or you can follow Molsoft on the following websites. We'll also place a recording of the webinar on YouTube. In this webinar, we'll learn how to make slides and add smooth and blending effects to your molecular display, how to make molecular documents, that is, you have a panel here with text which can link directly to 3D structures, how to download ActiveICM and embed fully interactive 3D structures into PowerPoint, and how to make a fully interactive 3D molecule web page using ICM Java Script. So the products you need for today's webinar are free downloads, ActiveICM and ICM-Browser. ICM-Browser will make the 3D molecules. ActiveICM will enable you to embed into PowerPoint. For the web, you do not need any plugin because we use a JavaScript and HTML5. So the technology is based on a single file representation, single file that contains multiple different data sources. So we patented this maybe a few years back, and the patent number's here. So it enables you to store sequence, images, plots, chemical data and other data, and text and 3D views, fully interactive text, so you can click on a hyperlink and it will go directly to the 3D interactive view. And you can store this in one single file. And then this can be then viewed in the ICM browser, which is a free download, or you can view it on the web using ICM Java Script, or you can view it in PowerPoint. It was developed with the Structural Genomics in consortium with the Structural Genomics Consortium, Brian Marsden's group there, and with Molsoft, Eugene Rausch, Ruben Abagayan, and Max Totroff. And you can read the paper here on +1. Other journals published research using the technology to build fully interactive 3D papers, basically, so the reader could read and then also investigate the structure inside the journal website. All the files you make in ICM-Browser can be viewed on the iMol view, which is an app for Android and iPad. You can download this for free, so you can have a similar thing. You can have text and hyperlinks, view structures, and molecular documents. It's a convenient way to share your data with your colleagues. So in today's example, we're going to look at serine protease structure and make slides of different views of the structure. Serine proteases are involved in digestion, clotting, hormone activation, and immune system. The key amino acid is a serine here, serine 195 in this example, and it's in a catalytic triad with histidine, 50-histidine, and aspartate. And they aid in the removal of the hydrogen from the serine, which makes it more reactive to react with the target protein. This example has an inhibitor bound here in yellow, and this is trypsin. So let's move on to that. So we open ICM. The web code, PDB code is 2PTC. So you can see the structure here. We have two molecules, the protease in blue and the inhibitor in yellow. We can make a slide, initial slide of the structure. We could maybe call it a global view. Click here. So we have a slide. To make a slide, you just click on this camera button, and the slide is here. So that means if you make any changes or anything at all, you can always double click and you go back to exactly what you had before. So we have one slide. Now it might be nice to show a slide of the catalytic triad. So serine 195 is part of that triad. That serine is here. You can select it here by dragging, or I'm going to also select the histidine and the aspartate. So I'm going to right click here and choose select by residue number, and I know the numbers are 195, 57 and 102. So now you can see that the residues are selected here, and they're also selected in the ICM workspace as green crosses. So we can make them, if we go to display tab, anything we click on here will propagate to the selection that we've made. So we have now labeled them. We could undisplay the inhibitor. We could also label the residues, color by, choose color for the residue labels, and then we could zoom in a little bit like this onto the catalytic triad. Okay, now it might be nice to look at the pocket in the steering protease. One way we could do that is to build a surface of the protein. We can do that by going to meshes and then choose plain solid, and that builds a surface like this. So we could just undisplay the CPK pointing up. So we can make a slide like this if you wanted to. So you just click on the camera button. We could also paint the surface according to where the catalytic triad is. So we will just go to select neighbors by residue number again. Just go OK, and then you can see when I made the surface we have another object here called G_skin, and it tells you what it's formed from, to PTC, the E chain. So if we right click on there, we can say color by atom selection and then choose red. Okay, so that could be another slide if we wanted, and maybe we could show another slide with the inhibitor bound. Okay, so we have constructed 5 slides. Now we can add some transitions between each slide. If you right click on a slide, you can say edit slide, give the slide a name, so this is the global view, and you can add smooth transitions between each slide and also blending, so when you change its representation it will blend between each one. So you check there. It's going to change the length of the transition and simulation. I'll do the same with this one. This is the catalytic triad, and we just click here, and some blending. Same with this one. I can't remember what this one was. I think it's the surface, so we can just say surface of protease, and then just check each one. Okay, so so now when you click on the slides, you see the transitions are there, and the blending. Okay, so now you can also add rocking into your structure. So maybe we'd like the global view to be rocking, or if you click and hold on this button, you can change the rotation or the rocking or the speed and range of the rotation and rocking. And if you want to overwrite a slide, you click and hold on the camera button and choose overwrite slide. So now we can save this file as a project, ICM project, and then we can embed it into PowerPoint. Okay, so now we open PowerPoint. So the key things we need to include before you can embed in PowerPoint: you need to install ActiveICM. You need to have the developer ribbon displayed. Sometimes it's hidden by default, so you need to go to File Settings and choose the option to display the developer tool menu. And then there's a button here called More Controls, and then if you click on that, you'll see a list of controls, and one of them is ActiveICM. So to embed, I'm just going to do that. I'll just get a clean session. So I go to the Developer tab, choose More Controls, click on ActiveICM, and then click and drag the size of the window you want for your example. And okay. So now if we go to slideshow, and from current slide, this slide will then become active. So you can rotate using your mouse, so you can zoom in and out. You can make selections like this and center on those, so you can answer questions from the audience. If somebody has asked a question about the particular part of the protein, you can select it. You can also toggle through the slides. If you click on here, you get different options. Anaglyph view, so if your audience has 3D glasses on, they can view in 3D. You can add some rocking, you can change the different structures displayed that are currently in your ICM file. Okay. So that's how to embed in PowerPoint. So now introduce the ICM JavaScript. ICM JavaScript is for embedding 3D objects inside HTML web pages. It's a JavaScript port of the ICM shell and 3D graphics. It's compatible with all modern browsers. It does not require any additional installation. And there's full access to the ICM scripting functionality. You can view it on the mobile devices as well. And it has the same quality graphics as the regular ICM browser desktop application. So here are some examples. So if you just make a slide and import and export it as ActiveICM, ICM JavaScript, you will get some window like this, something like this, which is pretty similar to the browser. You can rotate, you can translate using middle mouse, you can make selections using the sequence here. Then you can toggle here to change the representations of the molecule of the structure. You can change the coloring. That coloring is based on selection, so similar to ICM. Let's say color by N to C, which is currently is, and then you can say color by secondary structure, for example. Or you can save what you've made into Google Drive or locally on your computer. You can also add your own slides as well here as well, so you can store current view. Let's go OK, and then if we change this, we should be able to go back here and go to slides, and slide 1 is stored here. So just click on that and it will take us back to that slide. Another way to represent structures, you can have more than one window. So in this case it's set for 2 different kinases to be able to rotate them at the same time, but you can also you can remove the synchronization and you can just choose, you can have multiple windows, whatever design you want. Another option is to display, you can share tables. So this is a hit list of the of some docking results. So you can send this to your collaborator and they can view it on the website without any downloads or anything like that, any additional downloads. So they can just click and toggle through the structures here. Another example is that with the molecular document, so you have an HTML page on this side and you can use the hyperlinks to toggle through to different structures or different slides that you made in your ICB file. So here we're showing the hydrogen bonds with a metalloproteinase inhibitor. Okay, so how to do that in ICM. So we've already got our slides prepared, so we're just going to work on the HTML side. So we can go File, New, and choose the HTML tab and go OK. So that loads a blank page here. If you right click and choose Edit, you can start editing. So you can say protease, so you can make that into you can use the options here. It's just like a typical editor, so body text, or we can call that header, title, extra large. Then we can add some additional, we can drop down here, we can add an image. We go to here, there's an image button, you can search for images and load them, change the size. Okay. So now we can also add hyperlinks from the text to the slides that we made. So we can say global view of the serine protease with inhibitor bound. So if you click on view, then you can go to slide and then choose the slide of the global view, go OK. You can see the catalytic triad is shown here. So, and then you can link to that slide, to the second slide, for example. Okay. So you can also add other options. You can add an external link to an external web page. You can add an action, so you can write an ICM script here. You can do a variety of using the ICM command line to change things around, to bring in new objects or do things. It's quite open for you to play around with that. You can also add checkboxes. So, for example, we could say display surface of the serine protease, and then we click on the checkbox here, this button here, brings up this, and you can do special customization, but we're just going to undisplay the meshes, which is the skin that I made for the surface of the protein. So just go OK. So that means if we click here, it shows on and off like that. So you can toggle on and off. So there's different tabs here. So this is the view. This is how your HTML will look. This is the edit, how to edit it. This is the HTML source, and you can have a split view as well with the HTML here and the editor here. You can also edit inside here as well. So I'm just going to edit using this panel. We can add images, so it's going to turn the rocking off here. We can make an image of this. Remember for last week, the image buttons are here. We can say add image to album. This will save it in ICM. And so that means now if we go to the image button, we should find it in the drop down here. And that's quite big. We just want to make it small. 3 and go OK. So we add the image here. You also notice in the ICM workspace we have a new section called images, and these are all the images that we've loaded into ICM. Okay, so once we're done, we can save and exit. And now to export this to ActiveICM Java Script, we go to File and we choose Export as ICM JavaScript HTML here. And you've got the option to export a chemical spreadsheet, and you can use HTTPS. You've got to give it a file name. You can just keep it like this and just go OK. So now you should have in your default directory the latest documents. In Windows it's documents. So you should have an HTML file called webinar example. So Chrome and Edge, you need to make sure this HTML file is in the web server directory and on the web server, because that's the way they protect from it's a protection. But Firefox doesn't have that. The Firefox browser doesn't have that protection, so you can right click and you can just view it just directly from your local machine by going open with Firefox, and then you should see so here we have the 3D window, and we have the slides, so you can click on the hyperlink and take us to the thing, and you can go to the catalytic triad and toggle the surface on and off. Okay, so here's another example with a molecular document with it's got a number of different checkboxes. You can superimpose the active structure of this adrenergic receptor beta 2, superimpose the active versus the inactive. And you have the ligand pocket displayed, shown here, and residues. You can have these distances as well, and other data rock curves for the hit list, and things like that. So we also have this hit list that we can go through and toggle. So if we go File, we can export this as ActiveICM JavaScript. Click here and go here. And then you'll see You also have the tables here. You got multiple tables taken to the web page, and the toggle the ligands on and off here. So go back to the PowerPoint. So ActiveICM JavaScript is a JavaScript object which implements a single ICM shell and graphics window. So you just need to call the ActiveICM JavaScript here. It can be embedded in any place in the HTML code, and the API is compatible with the old ActiveICM that we used to distribute. The ICM shell and scripting language is fully accessible from within the web browser and can run locally on or remotely a remote ICM script, and can run any list of ICM commands inside the web browser. So example here, display CPK for the hydrogen, for het atoms, and this is a command to display stick for a sphere around a selection in ICM. You can also have multiple calls, and so multiple pages. So each ActiveICM can be controlled separately from the user interface and JavaScript, or they can be grouped for synchronous rotation. Thank you very much for your attention. Next webinar will be on protein structure and crystallographic analysis tools. Things like measuring distances, measuring atom angles, and looking at crystallographic data, bringing in electron density maps, contouring them, building biomolecules, looking at crystallographic neighbors, and such like. Thank you very much.